Hi everyone! For this module, we're going to examine the structure of DNA, which is the genetic material that is passed on from parents to their offspring. The genetic code of DNA is made out of 4 types of bases, which we denote through the letters A, T, G, and C. Every person's DNA has 3 billion bases. However, the specific sequence of or order of bases is different for each person. For example, while Sally has a part of her DNA coded with A, then T, then G, then C, Sam could have that same part of his DNA coded with C, then G, then T, then A. If we extend this to all 3 billion bases that we have, we can quickly see how each person can have their own unique sequence of DNA. The differences in the order of the bases in our DNA is what makes all of us unique and different from each other. The main purpose of DNA is to encode the information our bodies need to properly carry out their function. This is accomplished through genes, which are small segments of the DNA that encode for certain traits. For example, there is a gene for eye color, and within this gene, the specific order of A, T, G, and C bases helps determine what color your eye is. There are about 30,000 genes in our DNA. Now that we've learned some important background knowledge about DNA, let's take a look at how DNA is structured by building a model of DNA using common, everyday materials. The materials that you're going to need are 8 pipe cleaners and a pair of scissors. First, take 2 strands of pipe cleaners and intertwine them like so. When you're finished doing this, do this again with another 2 strands of pipe cleaners, so you have a total of 2 intertwined strands. Now, cut the remaining 4 pipe cleaners into equal sized, smaller parts. It's easiest to do this if we fold them all together and then cut them. Once you're done with this, you should have 4 piles of different colored pipe cleaners. The next step is to twist the smaller pieces onto the longer strand. You want to do this along the entire length of the longer strand. Try to keep the smaller pieces at an equal distance from each other. Once you're done, you should have 2 strands that look like this. The next step is to tape the 2 strands together by taping together the ends of the small pieces. When you're done with this step, you should have something that looks like this. Finally, take the structure and twist it to complete the form of DNA. You should end up with a structure that looks something like this. This is the structure of DNA. We call this structure a double helix.